In today's lecture, we're going to talk about transcription and RNA processing. Before we talk about transcription, in other words, the making of RNA from DNA, we have to talk about the differences between DNA and RNA. There's many similarities, but there's many differences as well. Both DNA and RNA are composed of nucleotides, so letters A, T, C, and G, except if you look down here, RNA instead of having a T has a U, so that's one difference there. The type of sugar is also different. So instead of having deoxyribose in DNA, RNA just has a ribose. And the difference is that basically that you have a hydroxyl group in RNA, but you don't have that hydroxyl group in DNA on this sugar. And that really leads to RNA being very unstable compared to DNA. Also, DNA is usually double-stranded. RNA is usually single-stranded. We'll talk about differences as we go through the lecture. The secondary structure of DNA is a double helix. RNA has all types of goofy secondary structures, loops, hairpins, etc. Because it's single-stranded, it can fold very easily. And then finally, as I mentioned before, since DNA does not have this hydroxyl group, it's very stable. Right? You can't really react with the hydrogen that well. But RNA degrades very easily because it has that hydroxyl group. Okay, we want to see the forest from the trees always, right? So as biology majors, we obviously need to know the big picture, but of course we got to know the trees too. And so this is showing you the central dogma of molecular biology. And the central dogma is something that's very core to genetics. So the central dogma states that DNA is transcribed to RNA, and then RNA is, the word you should add here is translated to proteins. There's three main types of RNA that prokaryotes and eukaryotes share. So this messenger RNA, this is sort of the big dog. This is the one that carries the code. So this is the one that's going to be turned into a protein later on. Then there's also other RNAs that are very important as well, but they're not translated into proteins. So there's ribosomal RNA, which is involved in translation. We'll talk about that later. And there's transfer RNA, which is also involved in translation. We'll talk about that later. So these last two RNAs are end products in and of themselves. They do things. Whereas the messenger RNA, I think of it just as it says, is carrying the message. So messenger RNA is something that's going to be translated later on into proteins. Eukaryotes have a whole host of other RNAs as well. They're all listed here. I just want you to know they exist for now, so don't worry about memorizing this list here. You can put a little X here if you want. But later on in the course, we'll reintroduce these one by one. So where does transcription occur? Well, in prokaryotes, since they don't have a nucleus, transcription just occurs in the cell, right? In eukaryotes, since they do have a nucleus, transcription occurs in the nucleus and translation occurs in the cytoplasm. That's a big difference here. So we say in eukaryotes that transcription and translation are separated by time and space. The fact that one happens in the nucleus, one happens in the cytoplasm, one happens first, the other one happens second. Prokaryotes don't have that separation, so there's no separation in time and space because there is no nucleus at all. And so as soon as a segment of RNA is being transcribed, right away before that strand's even finished, the front end of the strand, or the 5' end, is already being translated. So each gene that you have is actually transcribed off of one strand of DNA. It's something that's very interesting. So I'm not saying all the genes on a given chromosome are all transcribed off of one strand, but a given gene. So if you look at gene A, let's say, here in this picture it's showing you that gene A is always transcribed off the bottom strand, right? And transcription always occurs in the 5' to 3' direction in reference of the new RNA being made. So this RNA is 5' here, 3' here. This other RNA, gene C, is also being transcribed off the bottom strand. However, you can see that gene B is being transcribed off the top strand. But again, only one strand of the double helix is transcribed for a given gene, and it's usually always the same one. How was this discovered? There was an experiment that was done in an organism, and I use that word loosely, right, because it's a virus, so they're sort of the zombies of the biological world. Can't really say it's alive. But there was an experiment done with an SP8 phage virus. There are two parts to the experiment. So I would, on your slide, take your pen and draw a line right through here like this. Okay, so draw a line right through here like this, because this is sort of the top half of the experiment. This is the bottom half here. In the top half, what happened is they had bacteria growing in radioactive media. The virus infected the bacteria, injected its DNA, and the DNA basically right here, sorry, this is the DNA, not this. This is the DNA. The DNA, when it transcribed to make RNA, the RNA was radioactive. In other words, we say the RNA was hot. So here we have radioactive RNA in the top part of the experiment. just sort of put that on pause, leave that there. Okay, now let's look at the other half of the experiment. They took the same virus, the SP8 phage, and they took the DNA, they separated it into two parts. So they denatured it, they separated the double helix. One part they called the light strand, one part they called the heavy. You'll see why in a second. And then what they did was they mixed it with this radioactive RNA. And what they saw was this. They saw that all the radioactive RNA hybridized with one of the strands of the double helix, right here. That's why it's called the heavy, because it's interacting with it, forming a heavier complex. The other helix, the other side of the helix, or the other strand, I should say, of DNA, these blue ones, did not interact with the RNA. So see how it's floating in solution here. The fact that the RNA bound all to one DNA strand but did not bind to the other showed that the RNA was made from one of the strands of the double helix. And in fact, it was actually made from the one to which it bound, if you really think about it. And so that was showing that genes are transcribed off of one strand and not off of both strands. Now in this organism, if you really think about this carefully, there's a little problem here. But in this organism, in this virus, what happens is all the genes of this virus are actually transcribed off of one strand. So this virus is unique. It's different from what I told you on the previous slide. But this experiment, these results led to a later experiment that sort of discovered that, in humans, each gene is transcribed off of an individual strand. Okay, so subtle difference between the two. So transcription occurs in three phases. There's initiation, there's elongation, and there's termination. So let's talk about those. And we're going to talk about certain phases more than others. So it's just a little segue slide here, right? So showing you transcription happening. Okay, so transcription initiation in prokaryotes happens via this mechanism. So you can see that here's our gene, called an RNA coding region. Just upstream of the gene, we have something called the promoter. If you recall, the promoter is a sequence of DNA that says basically transcribe after me. So the promoter itself is not transcribed, but right after the promoter we have this transcription start site. The template strand is the strand of the DNA that's going to be transcribed. The non-template strand is the one that's not being transcribed. And so you could see here that we have transcription occurring, we get the RNA to transcribe, and then eventually it encounters a terminator sequence. The terminator itself is transcribed, but after the terminator is complete, it's basically encoding for the fact that you want transcription to stop. Okay, so that's sort of the whole scenario there, but how does initiation happen? So there's something called a core RNA polymerase. It's composed of different parts. There's two alpha subunits. There's a beta subunit, a beta prime. All those are required, right? So I just sort of note that. Then there's an optional subunit called an omega subunit. Now, it's an option, but you'll discover in a second that it's pretty much an option you want to have, but technically you could get transcription without it. The omega subunit, to me it looks like two little tires, right? And so I think of it as like holding the train on the tracks. In other words, the job of the omega subunit here is to hold the RNA polymerase on the DNA while it's transcribing. So if you didn't have it, you get transcription, but the polymerase would keep falling off and you get shorter transcripts than you should. Once you add something called a sigma factor, this is another subunit, then you get something called the RNA polymerase holoenzyme. So now we got to think, well, what's the job of the sigma factor here? So once it attaches, what the sigma factor does is it guides the RNA polymerase to the start site. So in other words, the sigma factor says start transcription here. If you didn't have the sigma factor, or if it was dysfunctional, then you would still transcribe, right? So it's optional, but you would start at the wrong site. So obviously it's an option that you want to have. Okay, good little question to think of. Which of these is required, which of them is not? Right? Make sure you can answer a question like that. Okay. If we look at transcription initiation further, we'll see that the promoter has certain sequences called consensus sequences. And what consensus sequences are is they're sequences that are similar in all organisms but not perfectly identical. So in other words, and this is in prokaryotes, but when we look at all different prokaryotes, they have a sequence that's almost like this, a sequence that's almost like this. And these recruit proteins like the sigma factor to this site to allow transcription to start just after it. Now when we say consensus, what does that mean exactly? Well, it's the same thing as if we took a vote in our class. Right? So if we took a vote in our class, we might not all agree on what kind of pizza to order, but the consensus, in other words, most people might say, I want sausage on the pizza, or I want pepperoni, or something like that. So not every single person agrees, right? We're not all identical in our thought, translate it, they're not all identical sequences, right? But they're all close to that. In other words, the consensus or the majority wanted this option of pizza, or the majority on average would have this sequence. How do you figure out the consensus sequence? Well, it's actually very easy. So what you do is you line up a bunch of sequences, right? And you take these promoters, you line them up from different organisms, and you say, okay, what's the most common base at the first site? What's the most common base at the second site, the third site, the fourth site? So you can see here we have T, T, A, T. The most common is T. Here we have A, C, A, A. The most common is A. Here we have T, C, T, C. T and C are the most common, right? But T and C are both pyrimidines. And so you can see that here we would have Y, Y standing for pyrimidine. In other words, you need some pyrimidine. Doesn't matter which one, but you need some. Let's look at this row here. A, A, G, G. You could have A or G, right? Equal probability. But A and G, pure as gold, right? A and G are purines. So R is the code we use for purines. So any purine could be at that spot. Here we have A, T, C, G. N means any base could be there, right? Any base could be there. Here we have G, C, G, G. There's actually an error in your slide, in your textbook actually. Change one of these G's to a C. So if you did, then you would have half G's, half C's, and you would say C/G, right? You can't use Y or R because they're not both pyrimidines, they're not both purines. But that's how you do a consensus sequence. So eukaryotic polymerases are divided into different categories. Each category has its own, its own duty, if you will, right? Its own duty. You should look at these and just sort of, I would know this grid, I would know this chart, I would know which polymerase encodes for which type of transcription. Okay, transcription elongation happens. We're just going to say it happens and, leave it at that, right? But let's focus on termination. So in termination, the transcript's growing, growing, growing. Eventually, we're going to stop transcription. So how do we know how to stop? Well, the polymerase is going to encounter two inverted repeats in the DNA sequence. So you could see two inverted repeats. After that, they encounter a sequence where there's a lot of A's in the DNA, a lot of A's. And this is what happens. So these two inverted repeats will end up causing the RNA to hybridize to itself, to form this hairpin, because the hydrogen bonding is perfect between these sequences. They're complementary to each other, right, because of the inverted repeat of the DNA. So you get this very bulky RNA hairpin. Then what happens is you'll have here a string of A's, a string of U's. There's two hydrogen bonds between A's and U's, just like A's and T's. Remember, C's and G's have three hydrogen bonds. So a string of two hydrogen bonds, although there's some strength, they're actually relatively weak, right, compared to like a mixed sequence. So what happens is when you get that bulky hairpin and that weak interaction between the U's and the A's, right, that causes the structure to fall off. So the RNA transcript falls off and transcription ends. The fact that you have this bulky structure and this weak interaction is what adds to that, that hairpin structure falling off. There's a bunch of different RNAs that could be produced. I want you to know the top three for now. We'll talk about the other ones later on. The reason I want you to know the top three is because both each ribosomal RNA, messenger RNA, and transfer RNA are found in prokaryotes and eukaryotes. So that's why I want you to know those three for now. To know this whole top portion of the chart, just the top three rows, because those are the really big players. The other ones we'll talk about later in different lectures. Okay, so again, just some things summarizing what we just talked about, right? Things that we just talked about with transcription in eukaryotes. Okay, the final thing I want to talk about is eukaryotic RNA is processed in three different ways. At the end of transcription, you get basically something called a 5' cap added to the 5' end of the transcript. The 5' cap is basically the letter G inverted, and so it's added for stability. It prevents the RNA from being degraded. Something called splicing occurs. Right? When you have splicing, what happens is you remove the introns. Introns are non-coding regions. Exons are coding. I remember this, out with the introns. So you're removing the introns. Or remember, you're never going to get rid of Exxon. Exxon like the oil company. So it's a little pessimistic. But, I don't really mean any intention behind that. Just, a way to memorize it. Then at the very end, at the 3' end, a series of A bases are added. It's called the poly A tail. And that also adds to the stability of the transcript. In other words, if something's going to be degraded, what happens is that string of A's gets degraded. There's no code in that. Why is it important that RNA is processed correctly? Well, this is why. This is an example just to show you. So there's a disease in humans called progeria. It's not really inherited, right? So it's just caused by a spontaneous point mutation. But what happens is it's a mutation in the protein that lines the inner nuclear membrane. It's called lamin A. And it's very interesting because it's a silent mutation. So normally a silent mutation does not change the amino acid that sequence encodes for, that string of 3 bases encodes for. So usually silent mutations are not a big deal. We're all walking around with, probably hundreds or thousands of silent mutations. Doesn't really matter. But the thing that we notice with lamin A is that although it's a silent mutation, it shouldn't, in quotes, right, it shouldn't change the amino acid. What happens is it introduces something called a cryptic splice site. So the RNA is truncated. It's spliced where it shouldn't be spliced. It's shorter than it should be, and the protein ends up being shorter than it should be. And the protein ends up having a functional group at the end called a farnesyl group, right down here. A farnesyl group is a functional group that basically guides proteins to the nuclear membrane. So when you have this protein that's shorter than it should be, and it has this farnesyl group, what happens is, as you look at this next slide, you get this type of defect in the nuclei of individuals that have lamin A. So if you look at a normal, healthy human, their nuclear membrane will have this nice little oval appearance. There's, some lamin A, so lamin A is in red, right? There's some lamin A in the middle, right? But, nice thin little oval appearance. As someone ages, the oval might get a little, misshapen, a little lobulated. But even if you looked at a 90-year-old, it would never look like this on the right. It never gets to this point. This is an individual with lamin A defect, right? So the defect known as progeria, aging before they should. Pro in Greek means before. Geria comes from the Greek word yetos, right? So it means like before you get old or getting old before you should. And you can see that all the lamin A, the protein is shorter than it should be. What happens is it's dragged to the nuclear membrane because of that farnesyl group, and you get this lobulated appearance. What's the negative effect? Well, lamin A is involved in global processes like DNA replication, transcription. So when you have this mislocalization of lamin A, you end up having major issues with these individuals. Okay, so that's the lecture on transcription and RNA processing. If you have any questions, please let me know. I'd be happy to answer them.